Thank you, Alexandro. Um, where should I start? There are so many starting points today. So first, welcome all of you to this afternoon session on day 2. Sorry to keep you away from the beach and the sea, but I promise to tell you more about them throughout my talk today. And one of the reasons I'm here is because finally, in our department, we wanted to bridge the gap between the theory and the experimental groups. And I was just speaking to Alessandro throughout his time in Luxembourg. We never got to say hello, but we come all the way to Cargèse to do that. So I don't mind doing that more often, I would say. Welcome everyone again. And my name is Anupam Sengupta. I've been in Luxembourg for last little over 6 years. And quite a bit of what I would be telling today covers the work I started a bit during my postdoc, but built up mostly during our time in Luxembourg. And I'm very happy to share today that I'm joined by, I would say, even 2 experts in this topic, Soumitra and Cargèse, both of whom are part of the active doctoral training unit, and they will be also presenting posters. So whatever I am not able to cover or not able to go into sufficient details, please feel free to reach out to Soumitra, Cargèse, and of course, I would be around too, should you be interested in discussing with me in person. Thanks to Massimiliano, Etienne, and all the organizers for inviting me over. We are in this very special summer school, Energy, Information, and Evolution in Biology. And I'll take a slight detour today to get you a perspective of what we do in lab in this context, but also not just in lab. Often it so happens working in lab, we forget the context of nature. And in our lab, we try to bridge also nature in lab in some sense. And so I'll give you slight short snippets of that and how we try to also contextualize our results in lab also in the context of what we see in nature. So broadly, what we will be looking into today are different classes of organisms. So these are microscale organisms, namely phytoplankton and phototrophic bacteria, and they are photosynthetic in nature. Photosynthetic meaning, I think it's a very fundamental part of, let's say, reaction network, the Kelvin cycle. I'll briefly touch upon that without going too much into details, but broadly the classes of organisms we will be looking into are phototrophic bacteria, marine phytoplankton, some of them are harmful algal bloom forming, filamentous cyanobacteria, and also some of the collective phenomena that they can produce coming together. We won't be looking much into the ecological ramifications of harmful algal blooms. Rather, what I would try to highlight is how, as active matter physicists, we can try to understand step by step some of the key rather qualitative observations people have found in the context of harmful algal blooms. For example, why they are so resilient, how they, despite very strong constraints in resource and light conditions, they are able to survive really long periods of time. And perhaps with the eye of a physicist, we will start to be able to get to the bottom of some of these long-lasting questions. So just to give you a context, this is a work which came out of my postdoc lab at the time, now almost over 10 years. But what it tries to highlight is the diversity, but also the huge population of microorganisms which inhabit marine ecosystems. So if you just take a drop of water, so we do not have to go far from here, you just hit the beach if you haven't yet done it yet, and just pick up a handful or a few drops of water, and be rest assured, if you bring them under the microscope, maybe next time we can also put a live microscope here, we will be able to see quite a few of microbes, and not just that, a huge diversity of them in shape, size, morphology, density, how they swim or if they do not, and so on. Now these microorganisms are essential for our own survival, not least because they produce oxygen, but they are also key in holding a lot of the biogeochemical cycles in place as we talk today. So just to give you a broad context, what you see over here, this is a 10 years animation which I took from the so-called MIT Darwin project, where the goal of the project is to understand evolution of phytoplankton density, phytoplankton concentration over time. So each of these colors that you see over here, these are actually false colors, but they represent certain phytoplankton types. So be it cyanobacteria, be it rhapidophytes, be it dinoflagellates, and so on. And what you start to see over here, depending upon which part of the globe you look, there is a concentration of certain broad classes of phytoplankton. I forgot to mention, please feel free to interrupt if you need to, yeah, so we can keep it interactive. So broadly what you see close to the poles, you have a certain class of photosynthetic microorganisms which exist. If you are coming closer to the equator, here you have different, let's say, photosynthetic organisms, specifically in terms of the pigments and the photosynthetic pathways that they harness. More so, what you start to see over here, that they form these very interesting eddy patterns. So these are simulated data, but based on real satellite imaging. So one can then assume that at the scale of these microorganisms, it's not as well resolved, but these patterns are typically representative of the ocean flows that these microorganisms are present in, readily suggesting where microorganisms or the ecological niche that they belong also is correlated, if not causally related, to the hydrodynamic environment they are part of. So inherently, microbes, like marine microbes, they inhabit a highly dynamic environment. And what I would try to, let's say, demonstrate in today's lecture is also some of our own efforts, but also broadly our collaborators and researchers elsewhere, that it's not just that microbes, photosynthetic microbes, are at the mercy of these ocean scale currents, but often in contrary, they can also shape and tune fluid flows at different scales, not just fluid flows, but also the biochemical environment around them. The other aspect, I don't think I have to nail it down very hard. Over here, we are all aware of the current shifts in the climatic trend. And now it's been a few years already that there was a consensus paper from some of the major, let's say, marine ecologists globally, who came together trying to emphasize the need to conserve and preserve phytoplankton. Not because, again, that they are life-saving, let's say, in terms of providing oxygen, but they are also one of the largest sequesters of the carbon dioxide. So they use carbon dioxide for their metabolic process, thereby giving and releasing oxygen. But also when they die off, they form some of the largest sinking, let's say, of let's say processed carbon dioxide, which then sinks into the so-called benthic floor, that is the floor of the ocean. The other aspect where, let's say, as a physicist, it starts to get very interesting. If you start scourging through the literature available, you see very conflicting observations. And the conflicting observations arise due to the fact that phytoplankton, marine organisms, they are quite diverse in their ability to respond to certain changes. So what we are currently lacking is a, let's say, generalized framework to understand how these microorganisms respond to changes in their environment. I will shortly hereafter come to the details of what we understand by environmental conditions. But broadly, let's say some literature would suggest that, okay, changes in the climatic conditions are detrimental. It's reducing the diversity, it's reducing the biomass of certain phytoplankton species. But again, on the contrary, there would be literature which propose that oceans are turning greener, and that might sound very positive because we all want to see green these days, but maybe it's not, let's say, as superficial as the title of this paper suggests. So one has to get very careful in understanding what is the causal relation between the factors where the phytoplankton or marine organisms live, but also in relation to other factors which are confounding in nature. For example, typically when we talk about climate change, we talk about changes in pH, changes in temperature. And what we often do not take into consideration that climate change is also changing the global ocean currents and thereby the local hydrodynamic conditions. When you take all of them together, this can have far-reaching, complex and non-linear responses as compared to just looking at one of the factors. So what we also try to do in the lab through very systematic approaches, bring in one factor at a time, but also looking at confounding, perhaps non-linear responses in the way these species adapt and respond to changes in the environment. One other aspect which I would like to highlight over here, if you look at the broad pool of microorganisms which inhabit natural environment like ocean, and the pool of microorganisms which inhabit in your human system. We all know in our body there are trillions of microorganisms in the form of what is known as microbiome, and specifically if you look at the classes of bacteria which inhabit human body as well as in oceans, there is quite a significant overlap. So one of the aspects that we are also trying to build up is understanding how do these microbes constitute what is known as microbes in one earth. Clearly, we cannot just study microbes in the context of ocean without including humans and other life forms, and vice versa, because in doing so, what we will be trying to establish is a more generic framework of how microbes respond, irrespective of whether they belong to a natural marine environment or they are part of human physiology and metabolic processes. So this is one thing. Again, this would be slightly at the backdrop of what we would be discussing, but this also forms the broader context of some of the research in our lab which we won't be considering today, which constitutes specifically work on bacteria and their conglomeration and how they respond and react to changes in environment, more driven from human physiological angles. How do we tackle these systems? So we are lucky enough to live in a world where we are all aware of the concept of active matter and, let's say, materials out of thermodynamic equilibrium or systems out of thermodynamic equilibrium. Who amongst you haven't yet heard of these terms in the last, let's say, 2 days since we have started? Okay, so I take it as a yes that you all are aware of this. So, and in fact, because we are living in this time and, let's say, place where we often end up discussing about active matter, but in various contexts. We saw some wonderful lectures, and I'm sure many more will be coming up. So broadly, if you want to look at microorganisms in the context of microbial active matter, that's what we do in my lab, we are looking into essentially length scales which are typically of the scale of, let's say, hundreds of micrometers, maybe in some cases up to millimeters when they come together in collective scales. But broadly, we are interested in so-called phytoplankton. These are typically, let's say, tens to hundreds of micrometers in size, or bacterial systems which are of the order of submicron to few microns in size. But in studying these systems, we also touch upon other forms of active matter, and by that I mean, let's say, chemical phases or chemical compounds which are either actively released or actively produced, actively generated, or even actively modulated by these microorganisms as they navigate their environmental conditions. And this also constitutes, although they are in much smaller length scales, so we are here now down to molecular length scales, but they are very much part of the active microbial matter that we were interested in. One of the key aspects of these systems is that they are able to elicit so-called emergent properties, meaning when we look at a single individual agent or an entity, they might have certain properties, but when you bring together not just multiple agents of the same type, or you let them play around with the environment in which they are inhabiting, they give rise to properties and phenomena which cannot be predicted immediately based purely on their own intrinsic properties. So these are emergent states, and I would be also touching upon some of these and means and methods to study these in the context of microbial active matter. So moving forward, when we started looking into these systems, of course, the path wasn't and still isn't free of challenges. And I wanted to just highlight some of the key challenges that we encounter on the way, starting from what is known as cell-to-cell interaction. So now one has to be very careful whether to study these systems in either isolation, so by meaning at the level of individual cells, or whether it's better off studying them at a population scale, then lowering the level of complexity and starting to look at single cells, or in fact directly starting off with community scales and then going down in complexity. So we are a bit opportunistic in that way. So we, depending upon the system and the questions that we have in hand, we sometimes start it off at the level of single cells, sometimes subcellular, even sometimes at a community scales, and depending upon the questions we ask, we then try to go up and down in the scale of complexity. The second point is their spatial organization impacts their physiology and behavior. And this is something which we will see often, whether it is in the context of marine ecology or in the context of human microbiome, how these species organize in space as well as in time impacts their chances of survival. And that's something very bluntly we will call as biological fitness for today's purpose, right? And so it's not just that cells decide to spatially organize themselves depending upon their environmental needs, but also there are gradients of forces of chemical, let's say, concentrations and molecules which make this organization firstly possible, but also make this organization biologically meaningful to execute some functions. Microorganisms respond, interact, and adaptively actively adapt, which means it's not passive colloids. By passive colloids, I mean synthetic colloids, which can be passive or active, but these are really living systems. So they have their own whims. Whims is a, let's say, a colloquial way to put that there are biological variation. When you look from one individual to another individual, no two individuals would be same, meaning we have to rely a lot on statistical tools and account for the variabilities that we see in our results. So it's very important whenever we are starting to look into real biological systems is that we have to distinguish noise from real results. So very often naively we might consider, oh, we are at the verge of a discovery, but next day we come do the statistics more rigorously and it might just end up to be just an artifact of some, let's say, either human or environmental factors we weren't careful enough. So this is one key difference compared to so-called synthetic active matter, where if you have steady hands, if you follow the protocol, and if you can follow the protocol, most likely you will get to reproducible results. Working with biology, it makes you a more patient person. That's what I tell my people. So if you need some, let's say, key takeaway from your PhD time as a biophysicist, this could be one thing, that you learn to be patient, and most likely you will have to do your experiments 3 times over than just repeating it once. And when I say 3 times over, we typically do our experiments, and all the data that I will be sharing today, at least they have 3 biological replicates and 3 technical replicates. Maybe it's better I quickly explain what we understand by biological replicates. So biological replicates, you start with biological organisms, you grow them at a certain condition, and day 2 you again start the experiment new from, let's say, different organism which has no connection with the organism from the previous day, and each of these would be counted as individual biological replicates. For example, if you are doing an experiment with a phytoplankton species, so typically what we do is we would grow and propagate this phytoplankton cell every 2 weeks, because They live in a continuous culture, meaning they need to be constantly fed with nutrients, certain trace elements, certain vitamins, and so on. And so you have two choices now. You have a one set of cells which are growing in this propagation culture, and you can do multiple experiments from this same culture. So for those of you who are not in biological experiments, these would mean these are the technical replicates. So you have one culture tube, multiple replicates. But imagine you have 3 culture tubes, each of them biologically distinct, same organism, same conditions, and so on, and these would be considered as biological replicates. So that means a variability in your results and system can arise due to intrinsic biological variability across the 3 culture tubes, or they can also emerge due to variability across individuals or across populations, across individuals which constitute a population within a culture tube, right? So we account for both variabilities, and based on that, then one has to do statistical tests to really get to control versus meaningful results. So often it so happens that one has to be very careful about the environmental conditions, and which then has, let's say, cascading effect on the so-called secondary metabolites. So these are biochemicals which cells exude. We hardly have any control. We have control if we know, if we want to do some targeted experiments, but broadly we do not fiddle with them, because it's like a like an alchemist's, pot. And but these are extremely important, because without these secondary metabolites, the cells might not perform, might not be biologically as viable as they would be without this secondary metabolite. So this is something we do not touch generally unless we have a very specific reason to do so. Finally, in the context of today's talk, what I would try to emphasize a lot is that environments change, not just at the scale of climate change. So these are very long-term changes, so most likely the response is more evolutionary in nature, but also environments can change at ecological time scale. So again, one aspect to keep in mind, over what time scale the environmental parameters are changing. So broadly, we will talk about 2, sometimes even 3 time scales. So ecological time scales, evolutionary time scales, and geological time scales. So I think all of us are aware of evolutionary and geological time scales. So these are time scales which are much longer than, let's say, the specific division rate time scale. So for a bacterium, few hours could already lead to initiation of some evolutionary phenomena, but for a phytoplankton which divides every 2 days, for an algae which divides every 2 days, it might take few years before we really start to see evolution. So when environmental fluctuations happen at the time scale of division time scale of the cells, this is something we will be referring as ecological time scales, right? So you have ecological time scales, evolutionary time scales, and geological time scales. It's multiple evolutionary time scales. So something that we won't be touching upon. Then very briefly, towards the end of my talk, I would also touch upon aspects of critical transition. So I think Alex, who will be speaking on Friday, you will be going a bit deeper into it. We will be very briefly touching upon how one can transition from such single scale, individual scale phenomena to critical transitions that we are actually observing in nature today, be it, let's say, the breakdown of the microbial networks in ocean environments, or the, let's say, extinction of species. So these all represent different form and shape and arts of critical transition. And can we actually formulate a bioenergetic framework to understand the process of adaptation, the process of critical transitions that we observe in microbial active matter? Any questions this far? Yes. Yes, that's a very good question. These are photosynthetic organisms, so they have a daylight rhythm as well, and this is typically 14 hours to 10 hours. That's the cycle in which they have an intrinsic clock functioning. What we have observed also in our lab is if you expose them to permanent daylight, which you can, or you expose them to permanent nighttime, which is not very useful if you are working with photosynthetic organisms because light is an important source of energy. But if you expose them to permanent daylight, what you see is, very interestingly, they still continue their circadian signatures. It's dampened, it's reduced, the amplitude of the rhythm is reduced, but you still see the circadian trend. Okay. So overall, what we are talking about is a system which can be extremely heterogeneous, which leads to noise. Noise, I mean statistical noise in the data that we collect, but also there is quite a long range of length and time scales that we cover. And the overarching vision here is to try and understand if we can really scale the phenomena that we observe from the level of, let's say, subcellular all the way to community scale of these organisms. So we do this in the physics of living matter group in Luxembourg, and typically during the lunch I was very happy to interact with someone who identified as an engineer. So you are there, Ingrid. And so yeah, I was trained as an engineer as an undergrad, so I think there is no harm in saying that as an engineer you can become a physicist or even a biologist, right? But I do not know if I am either of them. I kind of mix the things around. And in doing so, we also apply, let's say, techniques, tools, even tricks from both engineering, physics, and biology. And this has been one of the, I would say, strengths of the lab, because when we are looking at the system, we are not just looking at the biological response, we are not just looking at the physical response, but what we try to offer is as much as possible a 360-degree view of the phenomena. Of course, it's not always possible to get at the detail of molecular resolution, but at least it's a combination of physiological and behavioral responses, and that allows us to create let's say a more holistic understanding, and in some cases even a causal understanding between physics and biology, specifically for microorganisms. One aspect that we have been also trying to explore, as you can see from the different, very diverse set of systems that we work with, they span from anything between microorganisms in relationship with, let's say, cancer environments, to photosynthetic algae, aquatic fungi, and so on. Our long-term vision is to also try and understand what are the general principles and what are the general, let's say, rules that determine the behavior, response, and physiology of these organisms. Another interesting aspect when you look at these systems is that often you encounter what is known as responsive biomaterials, because at the end we are studying real living systems which are being bombarded with different kind of environmental changes and fluctuations, and they offer very interesting insights as responsive materials. So there is a certain applied angle that one can also take upon or pursue once you are starting to understand these organisms from first principle approach. These days it's becoming more important to associate how your research fits the so-called the sustainability and climate development goals as enshrined by UNDP. And so broadly, just to give you a context, the research that we do, it fits some of the key sustainability and developmental, let's say, agenda and goals that has been set up by the UNDP. But mostly, let's say, the key that we are concerned is essentially life below water, climate action, and life on land. So these are the 3 key sustainability goals that concern us. So as I was mentioning, how do we tackle these systems across scales? So depending upon where we start, it could be at the population scale where we can understand and study the collective dynamics, but we can go then navigate either way. Either we scale up the process to study community scale complex ecosystem dynamics, or scale down to look at species level details, how the species behave at the level of single cells, and also often in subcellular scales. So often what happens between these scales, at the interface of these scales, that's where emergent properties really start to manifest. So when we scale things up from individual to cellular, individual to population, population to community, these interfaces are precisely where we are tracking some of the emergent properties. So quickly, this is part of my group, and as I mentioned, you might look out for Shoumitri and Cargèse today, who will be also having their posters. So broadly, in terms of techniques that we will be coming across today, it's a lot of the work we do is visualization based, which means we follow cells at the level of single cells, at the level of subcellular detail, but also at the level of population scale. So you directly start to see that it scales different length and time scales, and many of our experiments are based on different tailor-made microfluidic platforms. You can broadly categorize them as on-chip devices which we build in-house, but what we also do is we combine these with different characterization tools on chip, and so they include, of course, fluid flow and gradient generators, where we can finely tune gradients of temperature, gradients of let's say chemicals and so on, but we also include different visualization tools, including confocal, laser trapping techniques, atomic force microscopy is something we use very extensively, and more recently we have been starting to use Raman microspectroscopy and scanning electron microscopy. So broadly what we end up is a set of phenotypic data, so in the phenotypic space, which could be covering the entire biophysical properties, also microstructural data, which becomes very important. For example, whether a cell is able to change its shape, which then feeds back into their swimming motility, depends on how the cytoskeleton network of the cells are arranged. So that gets us to the microstructural data, something we try to do using SEM and TM. And finally, we have the molecular data, which then allows us to concomitantly track molecular changes in this system. So taking them together, we get the full picture. Of course, full is a very relative term, but then let's say it gives us decent amount of data so as to create a causal framework, which we then model and go back to lab based on the predictions these models make, and try to check if these predictions are getting validated in experiments we hadn't done previously. Let's say in terms of the key tools that we often use, so 3D microfabrication, not just microfluidics, but also laser-based techniques, including laser cutting and 3D printing, is one of the mainstays of the lab. We use a lot of, let's say, home-built automation. So this is, let's say, small-scale robotics that we often build. I'll show you examples of some of these. And more recently, we are also getting into molecular analysis tools and data-driven modeling. So what these essentially lead us to is a bottom-up mechanistic approach. So these are some examples. I just wanted to highlight some of our other stuff, which I won't be touching upon today. And these revolve around bacterial systems. So bacteria is much more tricky to track than a phytoplankton, simply because of the length scales. So typically when we look at a bacterium, these are, if you look at the length scale, this is about 2 microns. So, and each of these cells, these are something between 2 to 4 microns in size. So we are able to visualize these cells over long periods of time, starting from single cell, but also growing up to small bacterial colonies. And once we have this data, it's very important because now we have the entire spatiotemporal coordinates of the cells as they grow and develop into biofilms. And these allow us to do quite interesting post-processing of this data. For example, here, how bacterial colonies mix, starting from 2 individual cells. Here we start to see how multi-layer bacterial colonies develop. And in fact, when these colonies develop, they also create emergent flow fields around them. And this is evidenced by, these are passive particles. So these are beads which are 2 microns in size, floating over a growing bacterial colony. If you watch closely, you will see there is a bacterial colony below it. And these colloids are held together by very strong van der Waals forces because of their size scales. And the flows are strong enough that this bacteria generates to break these colloidal rafts apart. So suggesting that although these are very dissipative in nature, the systems, they can still generate sustained long-term flows. So these are over time scales of up to a day that these flows persist. And our hypothesis is that these give rise to allow them to distribute nutrients under different nutrient constraints. Finally, more recently, we have started looking into bacterial colonies in the context of certain cancer types. These include colorectal cancer, but also skin cancer, lung cancer. These are all cancer types which have, it has been shown that microbes, bacteria specifically, are associated with their growth and development. However, there is no mechanistic causal understanding of how bacteria contributes to their growth and development. And so all of these taken together represent different aspects of living active matter, so out of thermodynamic equilibrium. And just to give you in a nutshell, these are some of our recent publications. And for today's talk, it would be mostly these papers that I will be touching upon. Any questions? Because now I will be really getting into the world of phytoplankton. Before we do so, if there is any question about the tools and techniques, and then we can change gears. Okay, very good. So, yes, the protagonist of today's lecture is phytoplankton. They are a key and a vital component of aquatic ecosystem. I always start with this when I have to introduce phytoplankton, specifically photosynthetic phytoplankton, that when I was in school, I learned that terrestrial plants produce most of the oxygen we breathe. It took me all the way till I started to do my postdoc to understand that actually they produce half of the oxygen that we are breathing. So for every 2 breaths that you take, 1 comes from marine microbes, photosynthetic microbes, and 1 comes from terrestrial plants. So their contribution, I do not have to emphasize, is immense because they really made possible life on Earth. And they are one of the largest sequesters of atmospheric carbon dioxide, right? Of course, when situations don't turn well, they also give rise to ecologically and in fact also let's say socio-economically detrimental harmful algal blooms. And one of the things we still do not understand is how blooms first occur, how they're able to survive for extended period of time. So right, it could be weeks up to months, but their collapse dynamics is sudden and very abrupt, right? So blooms take few days up to weeks to form. They will be stable like a sheet, like a layer of very high outgrowth of algae in various ocean environments. And but when it comes to the time of they disappearing, it's a very sudden collapse. So something like a catastrophic failure, but there is no biophysical understanding of this process, and this is something which we are trying to understand closely. So these are just some of the examples of blooms in different conditions. So as you see, this is this so-called formed by red tide-forming algae. It's called Heterosigma akashivo. These were pretty dominant during 1970s, 80s on the coasts of Japan. Japan being a very, let's say, fish-dependent economy and seafood-dependent economy, it created quite a havoc in the Japanese economy. And that's when there was quite a bit of research going into understanding Heterosigma akashivo, the kind of toxins they produce. So many of the toxins they produced are neurotoxic. They're not harmful as neurotoxics to human, but they're enough to create large-scale mortality in fish and various sea organisms, especially those living in the coastal areas, right? More recently, with the influx of nitrogen from, let's say, rainwater and also terrestrial systems, which ultimately end up in our ecosystems, be it lakes or oceans, that also has given rise to quite periodic and extensive algal growths, especially also in inland waters. So it's not unusual to find these days that lakes and rivers, as well as coastal areas which were otherwise very popular tourist destinations for swimming, are getting increasingly infected or inflicted by these harmful algal blooms. One of the other aspects of this is when you have this massive outgrowth of these blooms, they create a big oxygen depletion zone in the lower layers of the water. And so that is not just the neurotoxins which create harmful effects, but also the dynamics or the equilibrium, the dynamic equilibrium of the gas exchange is altered for quite extended period of time before it can recover under natural ecosystems. Finally, this is not a bloom, but this is a very interesting phenomena called bioluminescence. Anyone? Yes, I see a few hands. So bioluminescence, it's a very interesting biomechanical response that phytoplankton, marine organisms exhibit a lot. So essentially, what you see over here, whenever the waves crash, that creates hydrodynamic to mechanical forces, which then lead them to create this bioluminescence effect. So it's kind of a, let's say, a marker for mechanical forces that they are experiencing in these local environments, right? So bioluminescence is also quite observed, widely observed in more complex aquatic life forms, and some of which are also being used as, let's say, futuristic responsive biomaterials which can produce light and as markers for stresses, biomechanical stresses in different environments. So the work I will be presenting today, it's mostly done by my postdocs who are no more in the lab, Francesco, Joy, and also Irvin and Orko, and PhD students Francesco, Narges, and Shoumitri. So Narges and Shoumitri, they're here today. So just to give you a context of the huge diversity of phytoplankton, so now it's about 10 years or even a little over 10 years, there was this so-called big, let's say, sampling event. It was called the Tara Oceans. I would invite you to check out this edition of Science. And what they did was, actually it started in France, and it's a schooner, it's a big sailing boat which went across different stations across the world for over a year, collecting ocean samples at different depths. So overall it was a massive exercise with a large number of research team involved, let's say, which was led by a French team. And what was very interesting, beyond the fact that what was very obvious, that there is a huge diversity of morphology, motility, the so-called phenotypic traits that define these organisms, what was very interesting was that a significant portion of the microorganisms which were sampled, these were being discovered as they were being sampled. So there was no previous trace of these microorganisms whatsoever. And many of the deep sea creatures which got, let's say, attention later on actually were discovered through this Tara Ocean expedition. So I would encourage you to look that up. It's quite amazing that I think we know more about space today than we know about what's below the oceans. And with that kind of a diversity, physicists have a field day to keep looking at different facets of adaptation, evolution, as well as responsive behavior that these organisms have. So when you start to make a phase map of, let's say, on one side you have the phenotypic traits, on the other side you have environmental factors, the combinatorial, let's say, exercise can be overwhelming. We are talking about not just a big data set, but it's a big, big data set. You had a question? So the traditional response about diversity is that it's evolutionarily determined, right? So the reason, if you remember one of the first slides where we showed this global map of phytoplankton, and you start to see that there's a broad category of organisms, combining or coming together in certain so-called ecological niches, either in the polar, on the equator, or in the subtropical regions. That's because of the existing conditions which have been set up over geological timescales. But what has been happening more recently, what Earth achieved over these geological timescales are getting disrupted. And so now, more than ever, these organisms are undergoing massive restructuring of their phenotypic traits, of their even genotypic traits, and of their, let's say, eliciting their adaptive capability. So the diversity per se emerges from the evolutionary reasons, but I think now we are starting to see how that diversity plays a role in allowing them to adapt, if at all, under changing conditions. Any other question? Okay. So as you can imagine, these phytoplankton, of course, do not live in isolation, not just at the level of a species, but they are also part of a bigger community, right? So because they are at the bottom of the food web, they are autotrophic mostly. Autotrophic meaning they can harness sunlight and convert that into energy. They are often grazed upon by higher organisms. So the immediately higher over them are the zooplankton. Unlike many of the phytoplankton, zooplankton are typically, let's say, millimeter or above in size. So essentially, in terms of hydrodynamic aspects, these are the inertia starts to play a role. Whereas when we look at phytoplankton, it's still in a Stokes regime for a good, for all practical purpose. And these phytoplankton, when they are eaten up by zooplankton, they also excrete fecal matters, which are essentially processed, let's say, phytoplankton, which are then also, that's one of the largest proportion of what is known as sinking particles. So these are also sometimes called biogels. And these biogels, they can again act as substrates for different marine organisms, zooplankton, and whatever is not processed, they eventually go to the bottom of the ocean. But what is happening more recently with, I'm very surprised with all this discussion about climate change, we are still allowing commercial companies to do deep water mining or deep sea mining. So this is very contrary, in my opinion, because we are essentially, with this deep sea mining, at the verge of releasing huge amount of carbon dioxide, which has been trapped and sequestered over geological timescales. And we are absolutely in a territory with no idea what the consequences would be, right? If we are reaching a tipping point, we would just be accelerating it. But that's my opinion. So broadly, when we look at, very simply, essentially if you have these green light harvesting organisms, it takes 6 molecules of CO2, combines with 6 molecules of water to give you 6 molecules of O2 and sugar, so glucose in this case, and in that process it also releases 6 ATP. But it's not just the autotrophic mode of life that they have, they can also exhibit what is known as heterotrophic mode of foraging, which means they can also feed on other organic compounds, or even in some cases they have phagotrophy. Phagotrophy meaning they can actually ingest other lower animals. In this case it would be mostly bacteria. So there are many phytoplankton species, although they have the ability to produce and cycle sunlight, not produce but recycle sunlight for nutrients and energy, but they, under certain conditions, they can also ingest bacteria, and this gives rise to their so-called phagotrophic way of life. And so essentially when you combine phototrophy, heterotrophy, phagotrophy together, sometimes it's also called as mixotrophy. So you might often come across phytoplankton species which are mixotrophic in nature. How is their daily life? Again, this is a very simplistic view, but this is a question which Alex has already posed. They have a diel rhythm. So diel means diurnal, over a period of 24 hours, depending again upon which ecological niche they belong, whether it's closer to the poles or closer to the equator. That would create some local variation, but broadly they execute what is known as the diel vertical migration. So typically, exceptions are always there, but generally when you look at the ocean and you do a vertical profiling of the ocean layers, the upper layers of the ocean are generally nutrient deplete. So there are not much nutrients on the upper layers of the ocean. It's also very stressful for microorganisms, especially the upper few millimeters, centimeters, because sunlight is bombarding UV rays, so it's extremely stressful. So it's unlikely we will find much organisms on the upper few centimeters. But as we start to go down, and if we keep going down, the lower regions, especially few tens to hundreds of meters below, that's where it's quite rich in the inorganic nutrients, so sulfates, phosphates. These are also being heavy molecules. They prefer to be at the lower levels, also due to density reasons. Now during the daytime, most of these organisms, when I'm saying most of the organisms, I'm referring to phytoplankton algae, so microorganisms. They have a tendency to swim against gravity, so go to the upper reaches to get sunlight to photosynthesize, and when the sun goes down, they come down towards the lower reaches, get nutrients, inorganic nutrients, and the cycle continues. Now you can imagine the migration of the predators of these organisms. It would be then phased accordingly, right? So if you have zooplanktons, then they would prefer to be towards the lower regions at night, because that's where the chances of getting phytoplankton are higher, and so they would have a reverse diel vertical migratory pattern. But it's not just light and nutrients that determine their fate, it's also the local hydrodynamic environment. And this local hydrodynamic environment, as you can also imagine, it varies from, let's say if you're close to the surface where it's dominated by surface waves. If you go lower, it's dominated again by hydrodynamic flows, which can be even much stronger, but these are due to upwelling reasons, so due to density differences, due to stratification, and so on. So taken together, it's essentially light, turbulence, and nutrients which determine the day-to-day fate of phytoplankton and how they perform, especially over shorter time scales. This is just a graphical representation how this is. So essentially you have the photic zone. This is the top 200 meters. That's where you will find most of the phytoplankton and zooplankton. Once you start to go down, this is where you start to get organisms which are highly evolved and more complex. But we will be just focusing in this region of the pelagic zone. In terms of length scales, if you look, so we are talking about bacteria and phytoplankton. So typically they range between submicron to about hundreds of microns in size. So that has hydrodynamic consequences, because if you look at typical Kolmogorov scale, so that's the scale of a turbulent eddy, the smallest turbulent eddy. So that is of the scale of millimeter. So that means when you see these large waves in an ocean, they keep breaking down into smaller eddies, and the smallest eddy that they break down into is the scale of about millimeter or a few millimeter. So for all good purposes, bacteria, phytoplankton, they are smaller than the Kolmogorov length scale, which essentially means what they are experiencing is a viscous dissipation. So viscosity acts to dissipate the kinetic energy, and then the cells experience a laminar shear over their body, right? So how do we mimic these settings in our lab? So typically what we do is we take data from ocean scale turbulence, if we go by the example of the turbulence, and this is one such data. So if you see turbulence signal, and this is time, and the way this data is collected, so there are a lot of publicly available databases where you can find passive, let's say, passive motion of different tracers in ocean scale, in ocean environment. And essentially we take these data and then program a chamber in which we fill up our cells, and then we run this chamber to simulate these ocean scale phenomena. Now, one thing to keep in mind over here is the dimensions of these chambers are so chosen that we are always above the typical Kolmogorov length scale, so that the cells still get to experience hydrodynamic environments which are relevant to them also in the ocean scale environment. To break down such simple turbulence signal, typically it's a superposition of two different, let's say, very basic signals. One is it's something like a box signal where you have periods of turbulence which are denoted by the thickness of these yellow bars and spaced by specific periods of no turbulence, because turbulence out in ocean is very intermittent. So they do not go on forever. They happen, then there's a break, and they happen again. And this essentially captures the strength of these turbulent cues. The second is the changing the rotation time. So rotation time meaning, let's say if you are looking at a turbulence chamber which is rotating like this, imagine a washing machine, right? It's undergoing rotatory motion. Similarly, imagine these are the frequencies of this rotation, and we could also then create different, let's say, strong and let's say soft frequency modes, so to say, right? So here the changes happen very fast, because if you look at on the time axis, they are changing very frequently. Over here they allow more time. It's a more gradual reorientation of the direction. So taken together, if we program our device, it gives us a phase plot where on one hand you have the rotation time, you have the waiting time, and then we have the phytoplankton response. In real life, this is how such a device looks like. So this is all done in-house. This is where exactly the cells are, but this whole mount, this is then programmed using a stepper motor, and this is attached to a camera, and this camera gives us access to cells swimming in these modeled ocean in lab environmental conditions. How do they look like? Essentially, this is a perspective view. So these are, again, one has control on the size and shape of these chambers. And when you look into these chambers, you can either visualize them in, let's say, bright field, or in this case, in fluorescence. So you start to see cells which are inside this chamber. So these are images and videos acquired from these chambers. So when you are looking at them at a lower resolution, but you get a large field of view, you start to see single cells swimming against gravity. So G over here will refer to gravity in all the slides hereafter. And what you start to see is, if you just let these cells stay in a chamber without any motion, they are just swimming against gravity. So let me remind you, these experiments are done in daytime, so meaning they are naturally tuned to swim against gravity. If you look at shine light of blue-green wavelength, since they are photosynthetic, they fluoresce in red, and these are the same cells which are fluorescing in red. One can zoom in further and study single cell, and what you start to also appreciate is that the cells are not just swimming up, but they also have a helical motion as they are moving up. Yes?
How do you explain that you show this arm of the protein? So effectively you will face a series for the same.
Sorry, I didn't catch the question.
So you show this arm that you also like. So this effectively creates a force.
Yes, yes, yeah.
So how do you determine gravity in that situation?
Very good question. This is, everything of this will follow. But just to give you a context, we take them as a solid body rotation. So the chambers are typically full, so we avoid any air spaces. So essentially we are looking at a solid body which is then rotating in this environment.
And also why is important the rotation? You said it's about the turbulence and create the sea light effect, but you said also that in their scale they don't experience the turbulence. So why is important then to have this turbulence?
Yes, that's a wonderful question. The reason we create turbulence is because overall we wanted to see what's the cumulative effect of changing the turbulence parameters in terms of linear shear stresses. Because when you have these different turbulence data and the results, what you can now start to do is you can arrive at what is known as, we call it the Taylor-Reynolds number, and that gives you the equivalent metrics of what kind of linear shear stresses these systems experience. But effectively what they are still experiencing is just linear shear stress. Yeah. Hmm? Yeah, that's again, maybe I should clarify this. In most of the experiments, when we are doing these in daytime, it's uniform light because we do not want to create any light bias. But at night time, you can also do these experiments, and in that case, whatever light is used, it's just to make the cells visible. And typically, we use a red light because that's where they are least photosynthetically active.
Have you tried different switching of the light on and off to see
Yeah.
the response?
Yes, so this is work in progress. Again, very good question. So today I will be just focusing on gravity-mediated swimming, but there is this, in parallel, this field of research where it can be also triggered by light gradients and light field, which is essentially photo-switchable. Yeah, please. Not in this one. Right, it's a closed system. The cells are as solid bodies which are being rotated inside this closed system. And the closed system is programmed as per ocean scale data we obtain from public databases. Yes, we'll come to that. Yeah. I think I need to pace up a bit. Okay. Very good. So this is when we are looking at a larger view. So you see we started at a lower level, lower portion of the chamber. This is a few millimeters. And you see if you release, then the cells are swimming up. And again, what essentially determines, as I said, we are not talking about light gradients, it's purely gravity mediated, meaning one of the main, let's say, understanding of gravitaxis instead in marine microbes is how mass is distributed within the cell. So it's a rather, let's say, straightforward analysis. So remember that the cells are micron scales. So what we are talking about is their swimming behavior. It's in the so-called Stokes regime. So they are perfectly torque balanced and they are perfectly force balanced. So there is no net acceleration or no net angular acceleration acting on the cells. Now if you have a cell which has a heavier bottom, and let's say for simplicity we assume that the cell is spherical in shape, and this kind of a body mass distribution will create an offset length. And this offset length is, if the mass was uniformly distributed, the center of gravity, or CW over here, would coincide with the center of buoyancy, or the geometric center, right? Now since there is a distribution of mass in a way that the bottom part of the cell is heavier, this creates the so-called offset length. It's called LW, which is essentially separation of the center of mass from the center of buoyancy. Now when you imagine if the cell is swimming vertically, it has the flagella. If you tilt it, it will have a tendency to reorient back to get back to its equilibrium position. And now since the cells are swimming with two flagella, this also has a propulsion that allows them to essentially swim against gravity. Now when we do the torque and force balance over here, we will get a bit detailed in the subsequent slides. What you will start to see is, maybe take this as an assignment tonight over a glass of wine, you will start to get to a relation which gives you the rate of angular rotation, from the tilted position to its equilibrium position. It goes as directly proportional to the sine of that angle. It's a very straightforward exercise one can do. So typically the trajectory looks like this. We take hundreds of such trajectories, look at the statistics, and then get to what is known as the reorientation timescale. The reorientation timescale is extremely important because it determines how gravitactically stable your cells are. If you have a low reorientation timescale, that means the cells can easily reorient towards its equilibrium and keep swimming. If you have a high reorientation timescale, it will be just diffusing around, so that its rotational diffusivity is higher. It would take longer time to find its equilibrium position. And so typically when we track the data, we get similar plots. We fit it with the sine function, and essentially the amplitude of the sine function gives us the time scale of the rotation, right? So this time scale tells us how vertically stable the cells are, and this is very key. Often we think when we talk about motile species that it's the velocity of swimming which matters, but actually there is a very significant role of the orientational stability, because velocity per se does not make them gravitactically stable. You had a question? Yeah, so we use TrackPy in many cases, and but often we tweak our program. So I would encourage you to speak to Shoumitri, who is in thick of things these days, getting a hang of his data. Yeah. But broadly, what you will see is essentially this is a single cell. We have trajectories like this. Then we smoothen these trajectories, and based on these, we extract essentially this fitting parameter over here. Yeah.
So these eccentric centers of gravity, are they under active regulation and trafficking? And if so, then how long does one value of this eccentricity persist?
That's a wonderful question. There are two ways I could maybe answer this. One is you can think of, maybe let me go back to the previous slide. They can regulate their center of gravity by internal reorganization of their organelles, and this is a work which has been done by Nargis, who will also present a poster, where we found that under nutrient-limited conditions, the cells produce lipids. And again, depending upon how far they are into nutrient limitation, they can very systematically determine where the lipids will be in order to tune their gravitactic stability and ecologically place them either closer to the ocean sunlight or closer to the nutrients, right? That's one way. But the other way that I will shortly come into, it's a nice way to segue into the next slide, is these cells can change their shape. Now, changing shape, as you may imagine, would no longer allow the center of mass and center of buoyancy to coincide with the hydrodynamic stress center. Under very symmetric conditions, if you look at fluid flow around these cells, the center of buoyancy, which is the geometric center, coincides with the center of hydrodynamic stress, purely due to symmetry reasons. They coincide at the center. But now imagine a situation where these cells are no more symmetric, I'll come to that, but they are more like an egg-shaped. This symmetry breaks again. And then there is a second length scale, which we'll talk about. So these were some of the early works where we were looking into how turbulence disrupts diel vertical migration of this gravitactic species. And what we did was, this was the initial distribution when there was no turbulence, no exposure to hydrodynamic perturbation. And we exposed them to movement of this, our home-built washing machine on lab. And what we started to see was, from a situation where most of the cells were at the top, it's expected that these are gravitactic, they started to show a bimodal distribution. And interestingly, so this is the control. We are quantifying what is known as an upward bias. So an upward bias of 0 would mean you have as many cells at the top as there at the bottom, right? So they are 50-50. If you have an upward bias of negative, you have more cells at the bottom than at the top. Whenever you have an upward bias of, this is quite greater than 0, there are way more cells at the top than there at the bottom. What we see when we do these experiments in the vertical plane, you see a statistical drop in the upward bias, suggesting there is proportion of these upswimmers which are now coming down, whereas when we do these same experiments in horizontal plane, so imagine you put your washing machine not vertically, but you have the horizontal on the horizontal plane. So now the rotation is in this direction, and you start to see there is no significant drop. That gave rise to two hypotheses. One, that first they are actually responding to this motion, whatever it is, when they are in vertical plane, and second, in vertical plane gravity acts but not in the horizontal plane. And so could gravity be also responsible for inducing this response? So we did many experiments to test, and what we found was that when we look at the top and the bottom population, we do their phenotypic analysis. You see, this is the vertical speed, this is their absolute speed. They were exactly the same on all metrics, suggesting that this split is an active split. It's not that they are settling because they are stressed or something else. And when we sample this top and bottom population, we start to get some first insights into why this might be happening. So first off, what you start to see, the cells at the top, they have a fore-aft asymmetry, meaning the cells curvature over here is somewhat lower than the curvature over here. Secondly, the cells at the bottom, they have a more or less equal curvature. But now you might also counter me, hey Anupam, earlier on you showed that the heaviest part of the cell needs to be at the bottom region for them to be upswimmers. Now what I am tracking over here, this is the cell contour, but this bright blob at the center, that's the nucleus of the cell, one of the heaviest organelles of the cell. And clearly what we see over here is, no, this is nowhere close to the midline. In fact, if at all, it's slightly above the midline. So that got us a bit thinking, in fact, are we starting to talk about another model of gravitactic stability? And indeed, it came up that when you have a fore-aft asymmetric shape, and then we did some fluid dynamic simulations, we took the data from our experiments, we put the flow field, and what we started to find was there was this another center which appeared. This is the center of hydrodynamic stress at which the drag forces act now, right? So now we have three centers. We have the center of hydrodynamic stress, we have the center of buoyancy, and we have the center of gravity. Now, depending upon how the relative distances of these centers and the offsets are, that gives rise to mutual competition. To get into a bit into the insight of how it looks like, what you see over here, you have a torque which is due to the hydrodynamic stresses about the center of buoyancy, and you have a torque due to the gravitactic reorientation about the buoyancy point, and these are now competing against each other. So the model that we developed, this is also, it holds good for spherically symmetric cells, of course. And what is very interesting over here is we are talking about length scales which are extremely small. So just about a micron or so redistribution of the centers of stress points is enough to allow them to restabilize either on the top or towards the bottom direction. Yes?
The dynamic center is like where is closer to the upper side, let's say, because the mobility in that size is bigger than mobility. So this is how you find the hydrodynamic center.
Yes, so what we do is we know the swimming speed of the cells, we know the shape, so statistically we extract the shape. Maybe I'll quickly run you through the next set of slides. So what we do is we extract the contours of the cell and we fit them using this curve. So we now have a very nice system of curves which nicely capture the cell shapes, and it also captures the cell asymmetry, which is with these additional factors C over A, right? So what you see, if you keep C over A 0, you get a set of ellipsoids, right? And if you change C over A, you start to increase asymmetry in the 4F direction. As long as you have asymmetry in this direction, your center of hydrodynamic stress goes above the center of buoyancy. When you tilt this, the center of hydrodynamic stress goes below the center of buoyancy, and that would have an opposite effect.
And something else is you are talking about the gravity and so this like gravitaxis thingy. But what I don't understand is, as you said, we are in the low Reynolds regime. So if principle of gravity doesn't exist at all, right?
Very good.
Because it's related to inertia, right? Related to mass. So how do they really experience gravity?
Gravity doesn't exist or manifest in terms of inertia, but it exists in terms of the torque it generates, right? When you're thinking of inertia, just looking at the forces. But think of it in terms of the angular forces, that the angular torque that they generate. And there gravity does play a role.
But torque isn't generated by a force.
Yeah, but the cell has a density which is slightly above water, so it will experience some gravity. Let's say there are no flagella. You leave the cell, it will have certain sinking speed, right? So it will reach a terminal velocity quick enough, but it will gravity, without the gravity, it will just be neutrally buoyant. Right?
So even though even in low Reynolds regime, we see sinking in this system.
In no Reynolds number, when they are swimming, they are countering the sinking, right? Because they are actively swimming also to stay counter the sinking effect. If you cut the flagella, which you can do chemically, then we can measure their, let's say, dead density. Yeah, so what I was showing over here, that we see this change in asymmetry, and this is related to then how these cells orient themselves in the vertical direction over a day-night cycle. And essentially what you can do now, depending upon the shape, I think the question you asked, where will the center of hydrodynamic stress would be, these blobs indicate the center of the nucleus. You start to see this line is the line where it is exactly balancing the torque, right? So if you have a cell which is highly asymmetric, but then its center of mass is above, it can still go against gravity. But if you have the other way around, the cell is bound to go down. And so that way we created a phase space. But of course, then the question was, why does it happen, and is it just for these species that we worked with? And that allowed us to really generically go out in nature. Luckily, those days I was at MIT, so we had access to sea, and we just went out and collected some cells, right? And what we saw was 50% of the cells responded to such a turbulent or such a hydrodynamic perturbation, suggesting that this was easier than, or it was more prevalent than previously thought. The question still remained why on earth they would like to do, and this something we at the time could not causally connect, but at least we correlated this with generation of reactive nitrogen species. So this is a stress marker, and what we found was the longer these cells are exposed to such turbulent cues, the more they were releasing stress molecules, in this case reactive nitrogen species. So what you see over here, as the intensity goes up of the fluorescence signal, it signifies the amount of reactive nitrogen species they are producing. So essentially what we are seeing over here is something known as bet hedging strategy, but not over evolutionary time scale. Remember, these experiments are happening over minutes to hours time scale. The cells divide every 2 days. So that means we are seeing bet hedging, but over a rapid time scale, so over ecological time scale. And this was at the time quite new to us, because any literature on bet hedging, so just a quick refresher of what bet hedging is, it's about you don't put all your eggs in the same basket, right? If you have stressful conditions, you need to have diversification of your strategies to handle stressful conditions, even if it comes at the cost of some sacrifice of some of the species, right? What is the sacrifice that these species are doing? Because some of the cells are deciding to not go against turbulence and swim against gravity, they are sacrificing their ability to photosynthesize. Photosynthesis is key for division of the cells, and that's why they are, this is the part of sacrifice they do, so that when conditions become more homogenized, they can go up in swim against gravity and then procreate again. But then this phenomena is happening at really short time scale, and this something was quite intriguing for us at the time. So this gave rise to another key question: is this really a generic stress marker? And so we started to understand what is the role of reactive oxygen species. And again, we got back to these experiments where we changed the turbulence properties, and we start to look at the turbulence response. And what we found was, compared to control experiments, there were two ways that we could elicit the response. Either if when we allowed them more time, they would take longer to elicit this behavior, or if we give them shorter pulses or shorter reorientation events, they would take more time, suggesting that there is a sweet spot of turbulence at which they give rise to this change in the properties. And this was then correlated with what we found again was this coincided with massive upregulation of reactive oxygen species in this case. And this we connected to the specific turbulence strength, and what we found was at turbulence strengths of 10 raised to about -4 to -5 in terms of turbulence dissipation rate, that's where the cells exhibit maximum shift in motility, and at even higher turbulence rate they completely get rid of the motility. Let me then just end this talk with a small insight into why did we talk about gravity at all, right? So if I go back to this curve where we have these vertical measurements, this is the control, and we saw a statistically significant difference between the red and the green, but not so between the red and the blue, then it gave rise to this hypothesis that does gravity actually play a role in this behavior that we are seeing. There's no way we can change gravity on Earth, so the only way we could do so was take them to a so-called parabolic flight, and as coincidence would have it, we had this flight above Corsica about 10 years back, right? So I do not know why the flight goes like this above Corsica. Maybe if you fall through, you will end up in water, and yeah. But whatever it is, so this was from the Swiss parabolic flight. This was our small team over here. And what we did was, we did simultaneously 2 sets of experiments. We had 2 boxes. Again, parabolic flights is not that easy to handle in the sense that we have very severe limitations of space and safety. So that's why we had to be very, I would say, conservative in what we could achieve. But what we made, or what we could take inside the flight, were 2 boxes, one which would continuously image cells which are swimming in these gravity changes, and an other box where the plan was that after every 4 rounds of gravity changes, we will fix the cells for transcriptomic studies once we have landed on ground, right? And typically, so if you look at the cycles, so we had cycles of 4 gravity shifts between about 0.1 g up to 1.9 g, and then we had 4 such cycles of 4 gravity shifts. And after every 20 minutes of elapsed time, we fixed our cells and then prepared them for the transcriptomic studies. What we found was, this is in a nutshell, is that there are 2 takeaways. From the motility studies, we found that actually a similar phenotypic shift or a similar phenotypic split was observed, like we saw for the vertical experiments on ground. But more interestingly, what we found was there were a couple of key, let's say, biomolecular markers which got upregulated in these transcriptomic studies. So one was this oxidative stress response, which again bolstered our angle that actually the cells, in absence of any physical organelle, might be actually using reactive oxygen species as a let's say detector of turbulence and gravity changes. That's number 1. And number 2 was that these cells showed an enhanced regulation or upregulation of lipid metabolism. So with this, I would like to just end my talk and leave you with a quick introduction to what Cargèse would be presenting in her poster, I think, tomorrow. So thank you very much, and I am looking forward to the discussions.